Okay. Um, I have to re-record this because apparently the audio was not recorded while I was giving the lecture. Quite annoying, but that's okay. Okay. Um, so today's lecture is mixing up three kind of interesting topics. I thought this would be a good way to end the semester. So I'll be linking microbiome with narcotics and how does that relate to the genomics. Obviously, in narcotics, I'm including alcohol because it used to be illegal and it is considered to be a drug. So I think this topic here is quite interesting, and by the end of this lecture, you'll be able to kind of understand how antibiotics can be used to actually treat cocaine addiction. Before I get started, and since we are at the end of the semester, I'd like to clarify a few things. First thing is on the essay, there's 5 letters that I'm using to guide my correction. The letters are I for introduction, M for methods, R for results, D for discussion, and then S for style and grammar. Obviously, sometimes in the essay, you'll find that my comments are really linked to the structure, but in reality, I do the thinking while reading your essays, in terms of critical thinking, in terms of logical flow, in terms of the arguments that you're using. So if you need more clarification on your grade, you can come see me and I'll tell you exactly what happened and explain those comments from there. So if you want to do well on essay number 2, I highly suggest you include um, those comments. The other thing is, if you see a positive comment, it just means basically include the thing that you're already doing well in essay number 2. For the midterms, you should get them soon because I'm quite done with the semester and everything I have to do. While you guys are presenting, I'll be correcting these things. Just one thing for the final, try to make full sentences instead of bullet points, because I need to see that you are able to explain the answer and argue and not just put bullet points. Here's this point, here's this information, give me the grade for it. Here's this information, give me the grade for it, etc. Instead, show me that you are thinking, and don't make me do the thinking by linking myself the bullet points that you're giving to me. The final exam is on Thursday, December 5th from 2 to 5 p.m. It will be in the Moslem Business Building on the second floor at room 2435. Those students who are with ACSD, I'll be seeing you guys as well. You can do your exam with ACSD. Because I have people looking that will be basically invigilating with me, then I'll be coming from one room to the other and vice versa until the exam is done. Last thing, or actually I have 3 more points. The presentations next week, this Friday, we'll be starting with Akil's team, and then we'll move to Alex's team. Try to send me your presentations a little bit before the lecture so I can make sure that they're ready. I'll be then posting these lectures for the final exam. Speaking of the final exam, so if you were in class today, you saw that I was not happy about one thing. Let me just reiterate this so that it's clear for every student, okay? I feel like I have been quite nice with most of the students and I have been quite understanding, and maybe that gave you the wrong impression about the goal of this class. So there are some students that ask me to remove stuff from the exams. So I have to this should be obvious, but I have to explain it. Let me deal with the exams, you deal with the learning, because your goal is not the exam, your goal is learning, okay? So every class that you've done at Concordia has about 22 to 24 lectures in it. And in no case there is a professor that is going to come and tell you, oh, you know what, I'm not going to include 4 lectures just for fun, There are actually people that are in here to learn the material. And so learning comes from practice, and that practice will be evaluated in the exam. So everything I said about the final exam, forget about it. This is the actual information that you need to keep in mind. And the information is there is 22 lectures that are included in the final exam. The questions could be about topics from the beginning of the semester, the end of the semester, or any of the presentations done by your fellow students. So you can read the papers that I posted on Moodle if you want. You can attend the lecture if you want. You can watch the recordings if you want. You just need to take into account all the 22 lectures, which means the 6 presentations plus the 19 lectures that I gave you myself. The only material that will not be covered in the final exam is the lecture on the Neanderthal genome, because we missed that class because I was not present. Other than that, the rest doesn't really matter. Just learn the material, come to the exam, and do the exam. If it's a little bit harsh, just remember that the goal of university is to learn and not to just remove things from exams. In any case, the presentations, try to keep them at 25 minutes because we want to have the discussion part after. And also we want people to ask questions. On the evaluation sheets for the presentations, you'll have to write a question from your own, which will be used by myself to count for your participation marks. Now, next week, we'll be also having the departmental evaluations for me. So that means I'll need a student from you guys to administer this and then bring all the sheets to the security. So you'll just get to evaluate me. Just remember to be professional and critical so that I can improve my teaching. And you can also go on Rate My Professors and also fill out the form there if you have the time for it. It will be greatly appreciated. Okay. So other than the serious stuff today, we're going to talk about an interesting topic. You probably, well, as I've seen in class, probably no one has ever heard of this information, which is where humans are considered supraorganisms, and we are considered supraorganisms because we do have our own self, but on top of us and inside of us, we have other organisms that are part of us, and this is why we consider ourselves supraorganisms. In fact, these other organisms are either fungi, bacteria, archaea, or viruses. So we'll just call them microorganisms from now. And actually, there's a lot more of them than there is human cells. So they're quite an important part of the human supraorganism, let's say. In terms of the set of these bacteria, fungi, virus, and archaea, we call them the collection of them is called the human microbiome, and it will be kind of the topic of today. And obviously, each part of your body has a different set of these microorganisms. So you have the intestine microbiome, the mouth microbiome, the skin microbiome, etc. In terms of numbers, because these bacterial cells and archaea and viruses are much smaller, there's about 10 times more of these microorganisms than there is human cells. And this is quite interesting because following the Human Genome Project, we were expecting that the 3 billion bases that the human genome contains will code for more than just 20,000 genes. But in reality, it does not. And this is weird because the variability that we have in our bodies when we compare each individual between us cannot be only accounted by 20,000 genes. 20,000 protein coding genes. In fact, Drosophila has about 20,000 protein coding genes. So this is quite weird. So there must be something else and that is contributing to this. And this is where the human microbiome comes in. As a matter of fact, there's about 3 million genes, protein coding genes that are contributed by the human microbiome. So this is quite big, which is actually 100-fold more than the, actually 1,000-fold more than the gene, the protein coding genes that the humans do contribute to the supraorganism. So there's about 10 phyla in the human gut microbiome. So today we'll be focusing on the gut microbiome because it's the one that's most known and the one that is kind of interesting for this topic. It consists of about 10 phyla, but 90% of the bacteria belong to 2 main phyla, so Bacteroidetes and Firmicute. The others belong to other phyla like Actinobacteria, Fusobacteria, etc. You can read a bit on these, but the major 2 groups or phyla are these 2 over here. So then the question comes of what does the gut microbiome do and how does it affect the function of the host? And this usually comes into different pathways. For example, it could enhance food metabolism. There's a lot of bacteria in your gut that do metabolize, things like, for example, lactate, etc. There's also the fact that these bacteria that are found in the microbiome are able to support a healthy barrier that defends you against the pathogens. So in your body, you have kind of a balance between commensal bacteria and pathogenic bacteria. And if you don't have the right balance, then you could allow for the proliferation of the opportunistic pathogens, which then will destroy the barrier that protects you from these toxic either organisms or toxins produced by these organisms. Another way through which the gut microbiome actually affects the host is by providing the host with essential metabolites. A lot of vitamins are produced by bacteria. A lot of sugars are metabolized by bacteria. Even neurotransmitters are actually the generation or the production of neurotransmitters is actually affected by the bacterial content of your gut. So the way you go from a healthy state to a pathological state is obviously by disturbing this microbiome. And so in the healthy state, what you have is a functional barrier, which is your intestine's lining cells with the villi here that have a mucous membrane on top of it. And then you have a set of bacteria that is in there. So these are considered commensal bacteria. The term commensal refers to microorganisms that will not harm the individual, but instead they will benefit from it, and sometimes they will benefit the organism from which they're taken advantage. So if you have an issue with the set of or the diversity of the microbiome that you have in your gut, then you can sometimes allow for the proliferation of these pathogenic flora, which then will break the lining of your gut. Because when you have these pathogenic bacteria that infiltrate your gut microbiome, or these opportunistic bacteria that take control, then what happens is that they will produce toxins and will recruit the immune system. And when the immune system is recruited, especially through the inflammatory response, it will actually destroy these cells that are supposed to act as a physical barrier between your gut and your bloodstream, your vascular system. And when that happens, toxins leak into the vascular system. There are also viruses, fungi, bacteria that leak into your immune system, and this could lead to sepsis, and it could lead to other effects downstream, pathological effects. So this is called dysbiosis, or the disruption of the biosis, that is the eubiosis that is found in your gut, and this can happen through different ways. Either you lose first way is through loss of diversity of the microorganisms that are found in here, and you lose the ones that are healthy for you. You can also have an increase in the pathogenic flora, and this is sometimes through contamination or through allowing an opportunistic microorganism to flourish, and they wait for you to be weak or your immune system to be weak to then appear, or also from the loss of beneficial microbes. And then obviously you can combine these between them. You can have a loss of diversity, and because of the loss of diversity, you have a loss of beneficial microbes, which then allows for the pathogenic flora to flourish. Now, there are many mechanisms that are proposed for dysbiosis to lead to disease. Obviously, the impaired barrier function that happens. You also have overgrowth of pathogenic bacteria, inhibition of protective bacteria. You have translocation of the bacteria into your bloodstream, and also the bacterial products. For example, gram-negative bacteria that is found in your gut has the LPS, the lipopolysaccharide, which is a toxin for you. And if you release that in the bloodstream, then you'll end up causing damage. And we'll see exactly how that happens in a second. Obviously, if you have these pathogenic bacteria, then you'll induce this immune response with the pro-inflammatory cytokine release. And as you've learned in immunology, probably the cytokine release is usually not good for you, especially if it leads to chronic inflammation, because that leads to tissue destruction. And sometimes cytokines are even shown to act as oncogenic small molecules. And the last one is the concept of leaky gut. So that's shown in this slide here. Technically, your epithelial intestinal cells are lined up very close to each other, and they have these tight junctions that keep them together, right? And you have a mucus layer on top of that, which offers a higher level of protection. And you have a flora or a bacterial flora that is kind of diverse, but contains some beneficial and some opportunistic microorganisms that are sleeping. The moment that you end up having, for example, a non-balance between these, or you end up damaging the lining of your intestine, then what happens is that you allow for the toxins and the pathogens to leak through. This is why it's called a leaky gut, and go inside your bloodstream. And obviously, you can imagine if you have bacteria in your blood, then you end up causing a lot of problems downstream. Now, drugs pass through, antibiotics pass through, stress hormones pass through. So you can imagine if you release the content of your gut into your bloodstream, that's not going to be a good thing. One interesting disease that I want to talk about, or actually two, gluten is found in bread, and if you have a gluten sensitivity, you usually have either the syndrome or celiac disease. As I told you in class, I do have the celiac disease, and basically when you eat gluten, you lead to your body undergoing, detecting it as being something external to you and not healthy for you, let's say, more like pathogenic, and your immune response to it is to have an inflammatory response. And so the inflammatory response recruits these immune cells that come in here and then just start destroying everything and breaking these tight junctions between the lining, the epithelial intestinal cells, and also by breaking the microvilli that are found on these cells. Now, two things happen. First, there's leakage of all the stuff that I told you about into the bloodstream, which is not good. But also, one interesting thing for this lecture is the fact that these epithelial cells produce many neurotransmitters. For example, the major place where serotonin is made is inside the intestine. So obviously, if you destroy all this, you're not producing serotonin, which is quite an important hormone or neurotransmitter for the body. Lactose intolerance is also the same story. You have casein that is detected from the milk, which is an enzyme in milk that is detected and leads to the exact same response. So to study the link between microbiome and narcotics and their effect on the brain and addiction and all that, we need to first be able to study the human microbiome. And the NIH created, after the Human Genome Project, a human microbiome project, which had the goal of basically understanding the link between the microbiome, human health, and disease. Because before, to be able to study the microbiome, we had to take samples, fecal samples most of the time, and plate them, and then try to make sense of all the thousands of different bacteria, fungi, and all that were found there. And some of them are not even platable because they require specific conditions, which makes it even harder to actually do. So following the Human Genome Project, we had the technology to sequence the microbiome, and so now we just extract all the cells, break them, and then sequence them, and then from there we can actually study the variability in terms of pathogenic versus commensal bacteria and their role directly with the host, which is us. So the Human Microbiome Project characterized communities in different parts of the body, including nasal passage, oral cavity, urogenital tract, gastrointestinal tract, etc. The 5 aims were as follows. They were trying to create a reference set of 3,000 microbial genome sequences. Just to give you a perspective on this, the Human Genome Project resulted in 1 reference set of our genome. So now they're trying to get 3,000 from all the different microorganisms. And they used the 16S ribosomal RNA as a sequence to basically do that. So the 16S ribosomal RNA is very interesting because it is conserved across in each clade. And so what happens is that each group of bacteria has their own 16S ribosomal RNA, and it's different between the groups, and it's highly conserved. So you can use it to kind of do a triage of all the bacteria and make sense of the complexity of the microbial community. This way, when you take all the sample together, the fecal sample, and extract all the cells, you can then map it to a specific, you can map each of the genomes to a specific clade. They also wanted to, obviously, you don't do this just for no reason. They wanted to understand the relationship between disease and changes in the human microbiome. But most importantly, and what's interesting for this lecture, is the fact that they developed new tools that allowed us to analyze different microbiomes within different contexts. And this is what I'm actually using today to talk about the link between microbiome and narcotics. Obviously, if you're studying humans, you have to think, you're doing research on humans, you have to think about the ethical, legal, and social implications. So this is one of the aims of the project as well. As a matter of fact, they found that between different groups, ethnicities, they found different variations of this microbiome, and you'll see an example today. So the workflow for microbiome genomics is very simple. You take fecal samples or whatever samples that you have to do. At that time, they were using fecal samples first, and then they started taking, saliva and things like this. You extract, you don't filter out the cells, you extract all the ribosomal RNA from there, and then you just sequence it. And then they were trying to, and then you would do some sort of search within databases to link which 16S ribosomal RNA you found in what amount to the different taxonomic groups. At the end, they found that actually this was not, so it did allow you to do some taxonomy profiling, but it was not representative in terms of disease, because the type of bacteria that is found in your gut is not sufficient to tell you what it does in terms of a healthy or pathological state. In fact, we have almost the same amounts of, those two big groups, the bacteroidetes and the firmicutes, but for some reason we react differently to things. And so this was not a sufficient approach. You had to do whole metagenome sequencing. And this is similar to whole genome sequencing, which was done by Craig Venter. You just break all the DNA, and then you do the taxonomy profiling using this set. And then obviously you use this DNA here to actually figure out the genomes of each of the unique microorganisms that is in there, and then you can do that to do some downstream studies for diseases and things like this. Specifically, if we look at it a little bit more in depth, you take the fecal samples, you break the cells, you extract the DNA, you fragment it, and this here you learned already in lecture 5, I believe. You build the contigs from there, and because you're using these contigs, then you can kind of assemble them and then link them to a specific microorganism. So obviously the sequences will be linked, so when you're going to break the DNA, you'll have DNA from different microorganisms, right? And then only based on the contig assembly that you'll be able to separate them in different organisms. Obviously, this is not perfect. This process is called binning. It's not perfect because sometimes you will make mistakes. You will think that a fragment is supposed to be in organism A, but in fact, it's coming from organism B. So this requires some sort of filtering mechanism and some sort of QA, quality analysis, which you kind of learned about in the Neanderthal genome project paper that I assigned to you. Down of this, you also have to do some sort of genome quality assessment using the N50, L50, and NG50 that we learned about earlier in the semester. But at the end of this, you end up with a lot of data, and that data can be used for a few things. So I will talk about these few things in a second, but first, the Human Microbiome Project is divided into two groups or two phases. The first phase was just to understand the community composition of bacteria and to have the genome sequenced of all the bacterial cells that were found there and try to correlate it with phenotype, but not so much. In the Human Microbiome Project 2, they actually had the advantage of the development of the omics technologies at that time. And because of that, they were able to do epigenomics, proteomics, metabolomics, and things like this of the sort. And with this data here, when you integrate it all together, then you can study the effect of the microbiome in terms of diseases. So in terms of the Human Microbiome Project, we looked at three different things. They looked at preterm birth, inflammatory bowel disease, and prediabetes. To keep it short for today, I will just focus on preterm birth, but you can read on the other two. So just a little bit of highlights from the Human Microbiome Project. So they were able to generate 2,200 reference genomes that were isolated from the human body. So now we have a lot of sequenced DNA from the different organisms that are members of this microbiome. And this data was processed. So the processed data is now available. You can go on the portal, the Human Microbiome portal, and find it. And you can actually read on this and try to, play with it and see what was actually discovered. So one of the studies that is part of the Human Microbiome Project Phase 2 is called MOMS-PI, or Multi-Omic Microbiome Study Pregnancy Initiative Study, which had two steps. First one, they took 1,527 mothers who were pregnant, and they studied them longitudinally by collecting specimen through time from different parts of the body, let's say, including the reproductive tract and the uterus, and they collected about 206,000 samples. So that's a lot. That's about 1,000 sample. No, that's about 100 sample per 120 sample per person, which is a crazy amount. And they looked at different analyses, including measuring the set of cytokines that are found within the microbiome. They did some genomics, proteomics, and metabolomics. They also looked at microbial factors, because in the microbiome you have a different set of bacteria depending on which microbiome is being studied. So you need to be able to figure out what types of bacteria are found there, and you do that through the 16S ribosomal RNA, but also through metatranscriptomics, genomics, and things of the sort. Then they also did an in-depth analysis, and they looked at 600 pregnancies to identify contributors to preterm birth. So preterm birth usually happens in the first or second trimester of pregnancy, and it's usually caused by multiple factors. In this case, they were able to link the microbiome to this. For this in-depth analysis, they looked at the cytokine profiles. They are interesting because the cytokine profiles will tell you if you'll end up having an immune response in your reproductive tract, in your microbiome gut, and if that will lead, for example, to a leaky gut or not, or to some sort of destruction of the fetus membrane, right? So also you need to know which species are found in there, obviously, so you repeat the same analysis that was found over here. So they found that actually you had two distinctive sets of microbiomes that were either going to lead to preterm birth or term birth. So as you can see here, as you can see here in this case, if you have a term birth, then you will have a set of bacteria in your microbiome that will be distinct compared to the other one. And usually what they found is that as time progresses and as you go through the trimesters, you end up enriching the amount of lactobacillus species compared to the other microorganisms that are found in there. So their proportions increase. While in the case of preterm birth, you'll end up having lower levels of these lactobacillus species. That's the first thing. And second thing, you'll end up having these microbiota associated, let's say, bacteria with preterm birth. So in other words, what I'm trying to say here is that there are these bacteria that are going to lead to preterm birth if they travel from the vaginal lumen to the uterus, right? And if they do travel there, then they will lead to an inflammatory response that happens early on during your pregnancy, which obviously lead to destruction of membrane, things like this, and the release of or the loss of the baby, more like that. The other thing is, and the reason why you want to avoid inflammatory responses in the uterus, it's because you want to expand the size of the epithelium in the uterus, because you want it to be kind of, let's just put it this way, that you want it to be comfortable for the fetus, okay? And so when you reach the third trimester, then you need to reinstate the pro-inflammatory response, because now you need that for the onset of labor. And it seems like when you have these bacteria that travel to the uterus, they will lead to an infection, which mediates the preterm labor, kind of, because it will lead to, instead of having the anti-inflammatory stage that is supposed to be here, you'll make it inflammatory, and you'll kind of initiate the labor, right? The other thing is the lactobacillus bacteria needs to be enriched because it has many roles that are related to a healthy pregnancy, including the ability to help in vitamin D metabolism. And my vitamin D metabolism, I told you to search it in class, but since I'm recording, I'll tell you what it does. It's important for decreasing the inflammatory response, or actually not decreasing, but modulating the levels of inflammation. And actually, vitamin D deficiency leads to problems with the immune system. And as you can see, the immune system in this case is very important, because inflammation usually leads to onset of labor. You want to avoid the bacteria that are going to induce this inflammation and this early labor. Obviously, following birth, when the mother gives birth, then you want to reinstate the initial microbiome, which is distinctive, which is the healthy microbiome that does not contain those bacteria that can lead to an infection and then preterm birth. So this was kind of, I said it in a weird way, so just to repeat, there is a distinct microbiome that will determine if you're going to give birth, if the mother will give birth, or if the mother will lose the baby. And usually this depends on two things. The first thing is these bacteria that will lead to an inflammatory action by the immune system that do travel from the lumen of the vagina to the uterus, and then induce this inflammatory response. So these cannot be there. That's the first thing. Should not be there. And the ratio of lactobacillus cells to the other bacteria should be higher as we progress towards the third semester, because they modulate the levels of vitamin D, estradiol, glycogen deposition, and things like this, which are important for the pregnancy. So it seems that we're able to link the microbiome to preterm birth and to a healthy pregnancy through these factors. So always when you're thinking about the microbiome and its link to function, you always have to think about the immune response, because it is one part through which things get worse. You have an inflammation response, and that inflammation response, in this case, leads to an early onset of labor, which is not good. This human microbiome project actually ended up resulting in a lot of data. There were 30,000 samples that were, or 32,000 samples that were taken from many patients. And then we have different sets of data for each of the sample, including proteomics, cytokine set, metabolomes, lipidomes, and things like this. And this is all online, and you can go on there and see it. And you can use this, at your own interest to actually, whatever, be creative and play with it, and you'll probably have some fun with this. So now that we understand how we're able to study the microbiome in link with the disease, which is basically studying the set of different bacteria, their different concentrations, and then also studying their genomic structure, which tells us about the genes they have, the variants of the genes they have, and their ability to metabolize things that go, for example, through the intestine and then release them back in the intestine, or their ability to destroy certain parts of the body, for example, the gut lining, or induce an inflammatory response in the reproductive tract. We have the tools to be able to do that. So then we can study it in terms of narcotics and their link with, let's say, addiction and human health. So I'm going to start with something that makes sense for everyone, because alcohol itself outside of the body is quite toxic. So if you take it in your body, obviously you understand that just the alcohol itself will destroy your intestine and will affect your gut microbiome. And the alcoholic liver disease is quite a broad spectrum of diseases or syndromes, let's say, including fatty liver. Because you're drinking alcohol, the alcohol is metabolized and it's turned by the liver, and then if you take too much of it, you'll end up having steatosis, which is a fatty liver. Following that, it can get into the worst stages of steatohepatitis, fibrosis, cirrhosis, increase in acidity, and even liver cancer. So this is quite a dangerous disease, and our society seems to be kind of accepting alcohol as something normal. And in reality, it is not. It's actually, I would argue, it's actually worse than heroin itself. Although you don't see that, because usually when you see people on heroin, you see that usually they're either homeless or something like this. But in reality, alcohol addiction is as bad as heroin addiction, because they both lead to physical dependence, and they basically destroy everything, right? So we think that just because this one is legal, it's actually healthier, but no, it's not. Okay, so that's my little social awareness story. So be careful with this. Although like most of the heavy drinkers develop hepatic steatosis, which is an increase in liver in the fat in the liver, only a small portion of them advances to liver disease. So this is weird. If they're all heavy drinkers and they go through the stages one by one, how come they don't reach the advanced liver disease? And that's just because obviously the amount of alcohol that you consume is not the only contributor to alcoholic liver disease, but there must be other factors in there, potentially genetics, microbiome linked. And obviously today we're talking about the microbiome, so obviously this is the topic of today. But these heavy drinkers, usually they all have the same, or they have similar amounts of drinking. So how come, this 95% of heavy drinkers that develop steatosis, why don't they also develop this? And we'll see exactly why. So obviously this is the topic of today's class, so it's no surprise, but ALD, there's evidence that shows that the microbiome dysbiosis is a risk factor in ALD development and progression. Obviously, this is kind of a vicious circle where you have dysbiosis that leads to ALD, that leads to more drinking, and then you just keeps, going back and forth between these states. As a matter of fact, for people who drink, usually if you don't give time to your gut to recover, you think that you are okay, in the sense that, let's say, for example, you drink only on the weekends and once in the weekend, and you end up destroying your gut microbiome, which is kind of what happens in kind of this generation who are partying a lot. You think that because Saturday you drank and Sunday you were hangover and then Monday you went back to work and you worked the whole week, that you are not affecting your body because you're still continuing through life and things like this. But in reality, you don't give enough time for your gut microbiome to recover. And so you keep subjecting it to alcohol, and that leads to dysbiosis. And through time, this will lead to many problems, including, for example, anxiety, depression, and things like this. Now, obviously, I'm not saying everyone is going to go through this, but just a small link to this so that you understand where I'm coming from. Serotonin, which is the mood neurotransmitter, is made in the gut. If you have dysbiosis, you are not making serotonin. If you don't make serotonin, that you will pretty much be in a negative kind of mood all the time. You'll have increased levels of anxiety, increased levels of depressive behavior, and things like this. And also, alcohol itself is used as a anxiety relief mechanism. You think you're going to a party, but your body is bringing you to that party because of the anxiety relief that you will get from it, right? So this is things to keep in consideration. If there is dysbiosis, the neurotransmitters that are produced or regulated by the gut will be affected, and if they are affected, that's a direct link to your brain. Okay, so there's a lot of studies that are looking at ALD, and they've been focusing on the intestinal bacterial microbiome dysbiosis. They were also able to link the oral bacterial dysbiosis to to ALD as a risk factor. And it's true, because if in your mouth you also have bacteria, right? And if you're drinking alcohol, then you'll end up for sure affecting that microbiome. And if you affect that microbiome, you can have some sort of leakage into the bloodstream of the bacteria and go on from there, right? And obviously, for sure, if you're drinking alcohol, there are signals that are being sent on the gut-brain axis that will affect. Well, you do feel it the next day. Like, no need to actually expand on this idea, right? In fact, there are nerves that are linking your gut to your brain, and it's a two-way street, meaning that whatever happens in your gut is sent to your brain, and then your brain dispatches it to all the centers in the body, and all the hormone-producing glands and things like this. This is why when you drink alcohol, you have, for example, a withdrawal effect in the next day. It's because some of the neurotransmitters that are usually produced are basically disrupted. Their levels are disrupted, and this leads to the withdrawal effects that you experience. So, how does the gut microbiome lead to liver disease? So let's say you're partying, you're drinking alcohol, you go home 3 a.m, you try to sleep, and the next day you're pretty much not feeling very good. Well, what happens is that usually you even wake up during the middle of the night, because you guys already know. And so this is due to the fact that serotonin is messed up in your gut. This is why when you're like super drunk and you go to sleep, you don't sleep 14 hours straight. You end up waking up like 3-4 hours later, which is kind of weird because you've been up for 24 hours, right? It's just because you have disrupted the serotonin levels in your body and other neurotransmitters like GABA, dopamine, etc. So the alcohol goes in your intestine. It leads to a dysbiotic gut permeability increase, and then basically that means that the cells, the lining becomes weaker, and so you have leakage inside your vascular system. One of the things that leak is the lipopolysaccharide that is found on gram-negative bacteria. This lipopolysaccharide will travel to the liver and activate this Kupffer cells, and these Kupffer cells will produce all the inflammatory proteins or cytokines that you can think of, which leads to damage and which leads to liver fibrosis, etc. Right? As you can see here, the tumor necrosis factor, the interleukins, things of the sort. So alcohol, dysbiosis, endotoxins traveling to the liver, inflammatory response which destroys the liver and leads to liver fibrosis. How do we know that bacteria, that basically drinking alcohol affects your gut microbiome? Well, one of the things that told scientists or researchers is the fact that the pH changes in response to alcohol feeding. So there were these mice that were given alcohol, and then they were kind of studied throughout time, and they saw that basically if you give mice alcohol, at the end, they're compared to the controls, they will have different pHs. And one of the things that controls your pH is bacteria. Bacteria play a big role in controlling your pH because they play with lactate, they play with different molecules, acetic acid and things like this, which will change your pH. So obviously, if you have a change in pH in your in feces, that means you have a change in the bacteria that is found in there. And obviously, if you do sequencing and all that, you'll have a clearer answer. The same thing can be applied to oral bacterial dysbiosis. When you drink alcohol, you will affect this bacteria. You will allow some opportunistic bacteria to flourish. Just in that night, that's sufficient for them to do what they have to do. And then that will lead to toxins traveling through your lymphatic system or vascular system to the liver. And then obviously that leads to the same story that I just told you, right? This is just because of leakage between the different membranes. And additionally, actually that's it for this slide. So one of the way that we're trying to fix alcoholic liver disease is by restoring the gut eubiosis or the healthy gut microbiome. And we're trying to do this through many things, including diet modulation, probiotics, prebiotics, antibiotics. One of them that is kind of weird that you probably heard of is fecal bacterial transplant. So actually putting healthy feces, let's say, back in the body of a person that has ALD. Obviously, this is kind of quite weird way of doing it. There's also people looking at engineering bacteria and then putting it back in the body, kind of like a probiotic, but engineering the bacteria before using it as a probiotic. Now, when you're a heavy alcohol drinker, you end up with this disease. If you stop drinking, that's a good thing, but then you end up still having to deal with this. So these are very important to deal with this problem here. Additionally, there's a team that found that the gut microbiome modulates alcohol withdrawal-induced anxiety. So the way this is a vicious cycle, usually you have anxiety. You probably don't feel it or don't understand it very well, but usually the people that get anxiety end up kind of automatically using alcohol to relieve themselves from the anxiety, and then the withdrawal brings back more anxiety, and then the drinking, and it's kind of a vicious circle. Most of the people think that they are going to a party to just chill and hang out, but in reality, the body is bringing them kind of there to relieve this anxiety here through the impression that they're going to party and enjoy and whatnot. And so there's a team that gave alcohol to mice, obviously, and they played with a level of bacteria that is found in the microbiome through usage of antibiotics and whatnot. And they saw that actually the oral alcohol consumption was able to alter the gut microbiome, the composition, but not its abundance. So that means that the different sets of bacteria that were there changed, but the total amount did not change. And this just makes sense, right? There are some of them that are more proliferative under a certain condition, they actually can metabolize alcohol and others not. There's a bacteria, the enteric bacteria that was found to participate in the formation of alcohol withdrawal induced anxiety, and this is because the gut bacteria are involved in synthesizing and regulating neurotransmitters like serotonin, GABA, and dopamine. And if you Google these in link with your mood, you will see that serotonin is very important for your mood. GABA is as well very important for your mood, and dopamine is the reward system. So if you have these messed up, well, the next day you're not going to feel very good, right? So one thing that is interesting, though, is that there are scientists that are trying to use the gut microbiome as a therapeutic target for alcohol abuse associated symptoms. If you're able to establish the right composition of bacteria within the microbiome, then you can avoid this withdrawal induced anxiety, as well as the addictive behavior that comes with it, because it's a vicious circle, as I told you. Okay, so that's for a kind of a drug that is ingested and goes directly into contact with your gut microbiome. But what about something that doesn't even go there? How can it really have an effect on it? So cocaine, which is highly relevant in the milieu of, let's say, raves, electronic music, also a lot in the finance world. Actually, there's a lot of, high level, or let's say the elite, they abuse cocaine. So any high level profession was kind of found to be at higher risk of doing cocaine. So this is quite weird and interesting at the same time. But cocaine is able to induce the inflammatory gut by changing the mucosal barrier integrity. And so if you affect the mucosal barrier integrity, you allow for the microbiome to travel in the blood, well, part of it, and the toxins and things like this. So there's a team that administered cocaine into mice, which resulted in actually changes in the gut microbiome, which is quite weird. But you have to remember that the gut is linked to the brain through many nerves, and it's a two-way street. So if you're playing with the brain directly through cocaine, then the cocaine will affect your brain, but then it will also lead to will reply to every part of the body to which it is connected, which is the gut in part. And if you read on these drugs, you'll see that cocaine, actually, when you take it, it makes you feel like you're not hungry, even if you didn't eat for like 2 days. And this is just another supporting point for the link between the brain and the gut. And so taking cocaine that has a direct effect on your gut and the release of certain hormones, for example, for hunger, and things like this. Okay? So because of the advent of multi-omics and the ability to kind of sequence tons of data from the microbiome, then cocaine was able to scientists were able to show that cocaine mediates gut dysbiosis by upregulating some factors, the inflammatory factors, NF-kappa B and interleukin-1 beta. Now, this is just, you don't need to understand what the hell is happening here. This is just to tell you that we're able to link a drug to a microbiome dysbiosis through inflammatory mediators like these two. And in fact, we're even able to understand what pathways are potentially involved that are impairing the epithelial membrane permeability. And the people that were working on this found MAP kinase and ERK1/2, which are important for stress response. So in other words, you take cocaine, there is an inflammatory response that happens in your gut, potentially through the brain-gut axis, which calls in for these two inflammatory mediators, which then will act on the cells themselves and activate different pathways, which will lead to an impaired epithelial permeability. And if you impair this permeability, then you'll lead to other effects that we talked about a bit earlier. Now, there's a group of mice that were treated with a prolonged course of antibiotics to basically reduce the amount of gut bacteria that you have. And then by looking at these bacteria, it was interesting because bacteria with a reduced gut, hosts with a reduced gut microbiome, in other words, humans that were, or in this case it's mice, but even humans this applies to. So they were found to show enhanced sensitivity to cocaine rewards. So if you have a reduced gut bacteria, you end up behaving more, you'll try to seek more cocaine. This is quite weird, right? So this just shows another reason for why the microbiome is quite important for health. And in other words, if you take antibiotics, you disrupt your gut microbiome, and if you have problems with drugs like cocaine, you'll probably be seeking it more, as shown in this experiment. And it also showed that these animals had an enhanced sensitivity for the locomotor sensitizing effects of cocaine administration. If you see people that take cocaine, they're quite agitated, and if you're deeper into the addiction for cocaine, you'll see that sometimes they can't even stop shaking, and it's because their body has been sensitized to the actual, the repetitive movements that they do while they're agitated on cocaine. And so animals with a reduced gut bacteria kind of end up more agitated and more shake more, let's say. The other thing is so here I'm talking a bit in general because I don't want to get into the specifics of each one. Instead, I would just want to do a survey of all the different possible findings relating drugs to microbiome. So they were able to correlate these changes caused by a disruption in the microbiome to transcripts that are important for synaptic proteins in the brain reward circuit. Jeez, that was hard. In other words, we're able to link the behavioral changes caused by a change in microbiome to important proteins in the brain. And this is very interesting because now we can actually play with the microbiome to reinstate a healthy behavioral pattern and to decrease the sensitivity to the cocaine reward just by playing with the gut microbiome. This is crazy because people would think that everything happens in the brain, but as a matter of fact, no. It actually happens in the gut first, more than in the brain. Additionally, if you look at people that have cocaine use disorder, they're usually quite skinny. That's because they barely eat. And if they barely eat, they end up having a disrupted gut microbiome. And if you have a disrupted gut microbiome, you'll end up having an enhanced sensitivity to cocaine reward. And it's just a vicious circle that does not stop. So there's people that are actually looking at the use of probiotics to help these people that are they're kind of addicted to these drugs to heal from them. Because if already you're addicted and on top of that, you're making yourself more you're making yourself sensitive to the cocaine reward, which reinforces the behavior, then you're never probably going to be saved from this, right? So you need to reestablish a good gut microbiome, which then will reestablish healthier levels of serotonin, GABA, dopamine, which will help you in the efforts of recovering from addiction. The next drugs that we're talking about is cathinones or bath salts, and then amphetamines. If you watched Breaking Bad, methamphetamine is the drug that they make. These drugs are actually given to mice by a group, and these drugs all showed similar effects. They all changed the two major phyla, which is the firmicutes and bacteroidetes, and obviously these phyla have different species in them. So they played with the species ratios within these phyla, right? And they all had a significant effect on the microbiome. Now, I don't want to get into details. It's just to tell you that taking these drugs does affect the microbiome. Methamphetamine is smoked most of the time, so you can see how just smoking something does affect the microbiome. Across time, so when they treated these mice with these different drugs, across time, each drug led to a characteristic microbiome specific for that drug. So methamphetamine had a distinct microbiome, and then, cathinone had a specific microbiome, etc. Okay. Now, just a little bit of awareness here for the students. So in my time, there was kind of this epidemic of people using Adderall, Vyvanse, and all that to study. These are drugs that are given to people with ADHD, but obviously we're overprescribing for sure. I'm not saying that some people don't need it. A lot of people do need them, but not everyone, right? Especially those that use it to study, thinking that they're taking kind of the limitless NZT drug there, right? If you watch the movie Limitless, you'll understand what I mean. It does not work. As a matter of fact, it gets you in this state of disrupted microbiome. It affects your serotonin levels, and the next day you feel like shit. On top of that, the reward system is not working properly, and that's very important for memory formation and things like this. So if you do take these to study, don't do that. Last 3 slides. Let's talk now about morphine. So this one is quite interesting. So morphine is a drug that's given to decrease pain by the doctors. Usually it's given in advanced stages of pain. But also derivatives of morphine are used or abused in the streets. And morphine and its derivatives lead to production. So a derivative of morphine is actually heroin. And so there's this group of mice that were given morphine in an intermittent fashion but not sustained, meaning a similar fashion as a drug addict would actually use. So use, get high, withdrawal, use, get high, withdrawal. So this is intermittent. And they saw that this intermittent administration of morphine led to an increase in activation of microglial cells, which is a sign of neuroinflammation. That's step number 1, which leads to saying, do we have a brain microbiome? You can Google this for fun if you want to. I won't give you the answer here. There's also observation of hyperalgesia, which is sensitivity to pain. So this one here makes sense because you're taking morphine. It should prevent the pain from... it should decrease the amount of pain. But as you take morphine in an intermittent fashion, you'll end up being more tolerant to it. So you'll become sensitive to pain at some point. One of the other interesting things that happen is that you have an impaired reward response. And this is quite interesting because if you have an impaired reward response, that means you're less likely to want kind of the drug, right? And it does happen because if you look at addicts, they're at the point where it's like they taking it just because it helps them feel good, like get back to normal state because they're physically dependent on it. So they need it to function normally, but they don't really get the same reward from it anymore, and they become quite miserable, right? Then what they did is they used antibiotics to actually get rid or reduce the gut microbiota in terms of amount of bacterial cells. And weird enough, it mimicked the negative effects of intermittent morphine treatment. So microbiome disrupted by antibiotics is similar to giving morphine in an intermittent fashion. And there are a few things that were reproduced. Neuroinflammation because of the decrease of antibiotics. Because of the usage of antibiotics, it led to neuroinflammation. Without getting too much into why for this, there's a decreased effectiveness of opioid pain relief, which means your gut microbiome actually has a direct effect on the analgesic potency of morphine. So that means that if you take probiotics with morphine, it should improve its effect, right? because if you're using antibiotics, you're getting a decreased effectiveness. So if you use probiotics, you should have an increased effectiveness, right? The one that was kind of weird for me is the fact that when you play with antibiotics and you remove the gut microbiome, you end up impairing the reward response. So that's telling you that maybe antibiotics can be used to treat substance use disorders, because a decrease in reward response is very important. Because drugs, the way they work, you take them, you get a reward, and that reward is then kind of saved in your brain, and every time you will try to seek that reward, and you will keep taking the drug in higher amounts to get the same reward, right? And so if you use antibiotics and you impair the response to cocaine reward, that means this could be good for helping people that have substance use disorder. Now, obviously, I'm talking about the microbiome. The microbiome has cells that are beneficial, others that are not beneficial. So researchers, for example, could find the cells that were kind of affected by this antibiotic and that led to this impaired response to cocaine rewards. Just those cells. If we're able to identify those cells, those bacterial cells that led to this impaired response to cocaine reward, then we can design antibiotics that are specific to those cells and use them as a treatment therapeutic method to help people with substance use disorder. So this is some crazy stuff, and it's kind of the latest things that are happening right now in this field. Obviously, when the mice that were treated with the antibiotic were then had a restored normal gut microbial population, the neuroinflammation decreased and the negative behavior kind of was reversed. So then they were kind of back to their reward seeking behavior, which is quite weird. So I talked about the substance use disorder and the use of these probiotics and antibiotics in a very general way, because I don't want to get into the details. We don't have time for that. So it's just to give you an idea of how genomics was used to understand the link between the microbiome and, for example, substance use disorders. And you can find a lot of papers on this. I highly suggest to read them. They're quite interesting. One of the work that is happening in link to morphine administration is the fact that potentially probiotic therapy could be a safe and inexpensive way to prolong morphine's efficacy, because morphine's efficacy with time decreases as you become tolerant. So if you're able to restore the tolerance by giving probiotics, then you'll end up helping people, for example, that are in palliative care and running out of options of analgesics to remove the pain, right? And I think that's pretty much it. So I'll see you on Friday. Please send me your presentations slides ahead of time. I will be posting them so that students can study from them. As I said, 19 lectures presented by me, 3 lectures presented by you. All of that is to be studied for the final exam. Don't ask me what questions will be on the exam. Just study. Study the lectures, study the slides, study the presentations of the students, and that's pretty much it. Take care.